Welcome back, ladies and gentlemen, for the afternoon sections. In these sessions, we are going to present about EU regulations on veterinary medicines and medicated feeding stuffs. On these sessions, we still have two guest speakers who are the same with the morning, Dr. Claudia Loncansio-Benia and Dr. Gudrun Gauhoff to present us the topics of risk assessment of pharmaceutical active substances and medical feeding stuffs. And another topic is marketing authorizations procedure for veterinary medicinal products and medicated feeding stuff, which will be presented by Dr. Gudrun Gauhoff. ขอต้อนรับทุกท่านนะคะกลับเข้าสู่การสัมมนาในช่วงบ่ายค่ะสำหรับในช่วงแรกจะเป็นการบรรยายเรื่อง Risk Assessment of Pharmaceutical Active Substances and Medicated Feeding Stuff โดยดร. คลาวเดียนะคะและจะเป็นการบรรยายต่อด้วยเรื่อง Marketing Authorization Procedure for Veterinary Medical Products and Medicated Feeding Stuff โดยดร. กุรุณค่ะในโอกาสนี้นะคะดิฉันขออนุญาตมอบเวทีให้แก่ท่านวิทยากรค่ะ May I now take this opportunity to give the floor to our guest speakers Dr. Claudia and Dr. Gudrun, please.
Claudia, it's your turn.
After lunch, you know. Gudrun, so you start?
Yeah, okay. So I just wanted to check how much time do you give me? I just go through it, yeah? Okay, welcome to the afternoon. We will
So
now...
you come first?
Yes, we...
I see you change.
Yeah.
Okay. So we change. Audience, please. Change to Dr. Gudrun.
So, we have switched the subject around because Claudia figured it made more sense to do it this way. So I will start with the risk assessment for pharmaceutical active substances and medicated feeding stuffs. So this is now, we are switching into another field of closer together. Yeah. Okay. We are switching another field of EU legislation. In many states, it's feed additives and veterinary medicinal products or pharmacologically active substances used in veterinary drugs are handled in the same legislation. That is not in Europe. In Europe, the pharmaceutical legislation and the feed legislation have been developing separately, and it is only in recent times that we have more collaboration between these two fields. As Claudia already said, if there are substances which are used in both fields, then both agencies try to get together and find a similar approach. So you have to consider with a historical background that this legislation started in a totally different corner than the feed and feed additive legislation in Europe. We have, since 2009, a new legislation on pharmacologically active substances. Before, it was a legislation that was called Regulation 2377 of 90. We have made a lot of experience with that, in particular as that also meant that we had to go with the pharmacologically active substances used in veterinary medicinal products to go through a similar exercise that the feed additives go through now, because in the beginning we first of all didn't have a European legislation on this. The pharmacologically active substances that were used in veterinary medicinal products were only evaluated by the member states. That led to difficulties when food was found with residues to evaluate that. I remember when I worked in the Ministry of Health in Germany, that was in the late 1980s. We only had some regulations about withdrawal times, and we didn't have these maximum residue limits that we have today, and it created all kinds of internal market problems because nobody really knew how to address the fact that there were residues in there. So through the 1990s, after this 2377 was adopted, we went through a process similar as that now in the feed additives. All the pharmaceutical companies who had products on the market, they had to turn in a dossier to the European Medicines Agency, or EMEA as it was then called, European Agency for the Evaluation of Medicinal Products. It's been renamed EMEA, European Medicines Agency, since then. So it was then going through this process of looking at all of the substances that were used in medicinal products of veterinary use in the member states, authorized by the member states, to look at what was in there and to evaluate all the substances and, if necessary, to give them a maximum residue limits. This legislation has been repealed and replaced by the Regulation 270 of 2009, which is in many ways essentially providing the same principles, which are also the principles which are applied by Codex Alimentarius. So it's very much similar to the Codex evaluation. But we have also been appreciating more what is done in Codex, and will be also allowing Codex MRLs under certain circumstances to be more easily incorporated in this legislation. So what is this legislation about? It's about safety rules and procedures to establish, if necessary, maximum concentrations of residues of pharmacologically active substances permitted in food of animal origin. These are the maximum residue limits. And secondly, that is a new approach that is in this legislation, to also set procedures to set levels of residues of pharmacologically active substances in order to harmonize the control. Because we found out, even if you could establish that under normal use you maybe didn't need a maximum residue limit, there were certain problems arising when the substance was not used properly according to what is normally the case in veterinary medicinal products, and that we had problems with this kind of residue. And on the other extreme, we had problems with how to deal with very, very limited residues of certain substances which have been prohibited, to deal with them if there are only like very, very little contents in this in food. You might remember we had particularly also problems with Thailand and residues of chloramphenicol, very little, very few residues. And this approach came also from, or the problems were related also to the fact that residue control laboratories were able to find more and lesser residues of certain products, where you could also ask yourself, can this one molecule, 2 molecules, or whatever it was, really be so dangerous that you have to throw all the food away? So that's why we have established a more moderate approach to this problem of residues, even of substances which cannot be positively evaluated. That means which cannot be authorized for use, but where there might, to certain difficulties related to the growing of food, for instance, in aquaculture, where it is sometimes really difficult to get rid of certain residues, and you get then this minor residues to deal with that. So what is the link between pharmacologically active substances and medicated feeding stuffs? The legislation on medicated feeding stuffs doesn't really provide for an extra procedure of evaluation. It's more about mixing and good manufacturing practice for the mixed products. So what you have in medicated feeding stuff is essentially the same as you have in veterinary medicinal products. It is the pharmacologically active substances that are also evaluated according to this residue evaluation legislation. So Regulation 470 of 2009 is also the relevant evaluation procedure for medicated feeding stuffs. So what we're looking at in the risk assessment of residues of pharmacologically active substances used in veterinary medicinal products, which can then become residues in food, is the metabolism and the depletion of the pharmacologically active substance, if there is such a thing as depletion, the type of the residues, the amount potentially ingested by human beings over time without an appreciable health risk. And you see with this kind of words like appreciable how difficult it already shows that there is a kind of a gray line between this, which has to be often looked at on a case-by-case basis. We have, if there is no metabolism and depletion, then we have to have now, and this is new in this legislation, we can also take into account the monitoring data and exposure data otherwise available. This is something that is very important considering honey bees, because in honeybees you don't have a depletion. The bees just fly around collecting the pollen and putting it into the recipients that they have in their bee houses, in the beehives. They're not metabolizing it through their body, so you cannot speak of something like depletion. So we had to find an approach to deal with that problem in the bees. But normally, what you're looking at is at toxicological studies, which are carried out in laboratory animals, and then in the animal where the substance is supposed to be used. You find out what this kind of animal, for instance, cattle or pig, what kind of metabolism is carried out or is implemented in that animal. So normally it takes a certain amount of time, depending on what you're looking at, and that time might be different from whether you look at meat or whether you look at a product like milk or eggs in poultry. You get different kinds of approaches to the depletion. So from there you calculate from the no effect level, which is more or less a pathological threshold, where you don't observe anything in the testing animals anymore. You calculate from them via the depletion studies, with consumption figures which are very high and very from there have a very safe limit. What would be an acceptable daily intake, and from there you calculate the maximum residue limit. The consumption figures are basically the same that we use, is also the same that are used in Codex Alimentarius, so that's a global approach that you presume that somebody eats 300 grams of meat, for instance, a day. That's very high. I never arrived there, so that's already a sort of incorporated safety factor. From this model, you then have you use that in a scientific risk assessment, and you establish from that point of view the maximum residue limits. We have introduced, going a bit further than what is done in JECFA and in Codex and elsewhere, we have also incorporated some possibility to deviate from the scientific opinion in a certain way, going either restricting it more or making it more open, and that is depending on the availability of different treatments. So we have now allowed that it is possible to, even if a substance is somewhat critical, if there is no other treatment available that's under certain conditions, it's also possible to allow that a residual limit is established under certain circumstances, and the substance can then be used if there is no other treatment. So, instance, to avoid, it's possible to have an evaluation that is ending up on the positive list in the annex or in the list of pharmacologically active substances. That you can have substances there to avoid the suffering of animals if there is no other treatment. We have also, in line with what we have in the general food law, you heard that yesterday, we have the possibility to also consider other legitimate factors. So if there is technological aspects in food and feed production, for instance, if you cannot allow certain antimicrobials to be in milk because otherwise you cannot make any yogurt anymore, that is possible to consider that. We did that before, but not in such a structured way or expressed way. It's also the feasibility of control. That is also an issue. And there we are still discussing a subject currently, again on one substance, which is the problem of injection sites. That's you would say that if you use a substance at an injection site, it leaves quite a high amount of residues, but it would not be a problem for the rest of the meat. However, to set two different kinds of maximum residue limits, one for the injection site and one for the rest of the meat, would be very difficult to deal with for the control. So we are discussing that issue for years on end now, but we have come more or less to the conclusion now that it's not possible to have an extra residue limit for injection site because you wouldn't be able to really say, is this an injection site or is it not in the food control. We have also possibilities to limit certain use of substance to certain conditions of use. To also describe what would be a good veterinary practice if you need a certain substance. You will also be able to restrict a certain use if the substance is likely to be misused. And the key substance here has always been the clenbuterol. You can also see that from the Codex inclusion that has been discussed. It is a substance that is used in pulmonary disease in horses and also in cattle, and makes good sense there to use it. It has been overused in the past as a growth promoter and has also caused, for instance, in some food scandal years back in Spain, problems that people had who had indigested it. And you might also have heard of the bicycle riders who claim to have eaten too much clenbuterol residue meat, and then they got caught in doping affairs. So that is, the clenbuterol is still an interesting substance in terms of temptation of misuse. And we also can now consider in this evaluation officially the availability of alternative substances, as I said. This is really the lecture that we have learned from the rather strict 2377, where we ran into all kinds of troubles internally and also with other countries outside the Union on the use of substances. So we have still today the option of either after an evaluation to set an MRL, to set a provisional MRL, or to set no MRL, to connect all of them to certain conditions and restrictions. And the temporary use of MRLs is limited to a problem that is related to sufficient availability of data or lack of data, which can only be upheld for the longest time, 7 years. If then there is no data available, the substance would have to disappear. So we have circumstances after the authorization that you don't have MRLs for certain substances. If there is, we allow now, apart from the application of a pharmacologically active company or a interested party that is trying to market a veterinary medicinal product, we have now also a procedure which would allow the request of a member state if they see that there is no medicine available to ask for the evaluation. We have also the possibility for the Commission to ask for evaluation. If the Commission gets requests from several member states, that would be more appropriate to channel that to this. Or we could also accept an application of interested parties, let's say beekeepers or certain people who are keeping animals for which there are no residue limits have been established, to look into this to see whether we can set MRLs from this point of view. So you would either have to look at scientific data available, maybe also encourage scientific evaluations. You would have to look maybe at authorized uses in other countries, in third countries. So sometimes we find that in other countries there is more, there are animals, they keep other animals, like for instance in South Africa, they have more ostrich than we have, and they might have a product on the market that makes sense, and we can take that officially into consideration. We can also, what is important is the so-called use of the cascade. That is, if you use a substance or a veterinary medicinal product that's only authorized for one animal species, use it in another species, and under the supervision of a veterinarian, so you do that with the background of the veterinary science. And if this is overused, so that you see there is really a need, because the cascade is overused, which is normally supposed to be only an exemption, then you can also take this as a prompt to have a closer look at what are the pros and cons of using that substances. This is, however, only possible if there's a so-called minor and minor species or minor use. What is a minor species and a minor use is really not defined in union legislation, and we haven't been able to really address that. So we would have to consider also this as on a case-by-case basis. Personally, I think minor species is definitely not the right way of putting it. It's more minor uses, which is a combination of a certain species with a certain use. So, for instance, even if cattle is a species that is widely spread in Europe, you might have a need to have for a treatment of eye diseases, which is something that is not so often treated in cattle, because unfortunately you don't treat cattle so sophisticated anymore, because most of them get the amputation behind the head before they are really treated to a large extent. So you have minor uses also for this species, for which we have, which are largely used in Europe. So it's a bit a tricky term, this minor species. Why is it limited to this kind of terms? That's the other question you have to ask yourself. So if we would make that too widely available, we would undermine any research into the correct use. So you always have to sort of balance the research so that companies are interested in bringing something on the market for the correct species with the correct data and the studies behind it. And on the other hand, to have only available for these species where there is an economic interest in, where you have a possible turnover, that you only have medicines available for these species, because it only makes sense for the companies to invest into studies that are dealing with these species. We can also have now officially a view of the opinion, and we are doing that now in combination with another article in this legislation quite generally, because we have also included in the legislation an obligation for the agency to also consider whether a substance can also be used in I should say a residue evaluation can also be established for another species than the one that the applicant applied for, which we call extrapolation. So the agency has now all the time consider whether it is possible to use a substance where the applicant has only provided the substance, the material for, let's say, cattle, whether it can also be used in sheep and goats. And this is on the basis of the fact that cattle and sheep and goats have a very similar metabolism because they have the same kind of stomach divided into 4 different compartments, and so often from that point of view you can extrapolate the studies from cattle to these small ruminants as well. So we will always ask them to do that. It's not always possible because sometimes the metabolism is just too different from one species to the others. You cannot easily extrapolate all data from cattle to poultry because, as you can imagine, a bird has a different metabolism than a fish or a cattle. So we can then request a new opinion. So we hope that via this process, which has started 2-3 years ago, that we get more substances evaluated, and therefore we can address the problem of availability first of all, and also some of the problems that we have with imported products where residues can be found that of substances that we have no interest in Europe for, the industry in Europe doesn't have an interest, but who are evaluated elsewhere. So these are the 4 possibilities that normally come out of the evaluation. Either you have a maximum residue limit, and that maximum residue limit will be always allocated to a foodstuff or a tissue type in a certain species. You will have a provisional maximum residue limits if there's data lacking, and I must say the data that is most of the time lacking is the residue methods to find the residue in the foodstuff. So that is something that the pharmaceutical industry is not particularly fond of finding it out, because this method is afterwards anyhow used by the control laboratories, and they have no real interest, so we have to really often encourage them. So what they propose is often the method that they use only for finding the substance in the depletion study, but then you are looking for a substance that you know. So you know you have injected this XYZ, so you only need to look after XYZ. And many times for the control laboratories, the problem is to find that needle in the haystack. So to also find the substance if there is other residues, or if there is the substance is in certain tissue where it's difficult to find. So we have to often ask for extra studies in order to have a method of analysis which is really serving the purpose.